Next is the exhaustive search, and to understand the exhaustive search algorithm, we have to consider two examples. First example is that of restriction enzymes or restriction mapping. So restriction enzyme was first discovered in 1970, and the mechanism of restriction enzyme is that it breaks the DNA molecule at very specific locations. For example, we can see here one of the restriction enzyme is EcoR1, and it breaks the DNA at a very specific location. Whenever GAATTC nucleotides will appear in the DNA, that EcoR1 enzyme will cut that DNA from this location. So thousands of restriction enzyme has been identified and isolated, and generally mainly from the bacteria. So every restriction enzyme has its very specific site, just like EcoR1. If we have a bacteria or other organism whose sequence has been known, we can calculate the restriction fragments. Restriction fragment means the two fragment size from where the DNA is restricted by this restriction enzyme, and the fragments that are produced are called restriction fragments. So there are thousands of sites of every restriction enzyme in any of the genome, either from bacteria or from other organisms. For example, here we can see this is the double-stranded DNA starting from 3' to 5', but this is continuation because there is a limitation, we cannot write further on the right side. So this is the continuation of this double-stranded DNA, and we can see here the same nucleotides that are present GAATTC, and this is the complementary second strand of the DNA. Again, the same sequence is present here and here. This is a very short fragment of DNA. DNA is a very long molecule, and these fragments and these sites may be repeated many times in the DNA. The restriction enzyme EcoR1 will recognize this site and will break it in a very specific manner. This is the same DNA and the same sites, but here you can see it in two colors. So whenever the EcoR1 will recognize this site, it will cut this in a very specific manner. Whenever the site will come, it will cut it here. So for more clarity, we can see the next slide. So we can see here the same DNA, GAATCT, and when EcoR1 restriction enzyme will recognize this site, it will cut from here. So the two DNA will be, pieces will be separated apart. So in this way, all the sites will be cut in this manner, and small fragments will be created known as restriction fragments. So these restriction fragments are being used for many applications, and for using these restriction enzymes, either we can create a complete digestion. For example, we consider this as a DNA molecule, and these are the sites which will be recognized by the EcoR1, and EcoR1 will cut from these sites, and the fragments that will be generated will be here. Two will be of 2, one will be of 3, and another will be of 3 nucleotides, or we can say anything. So this is a complete digestion. So these are digested completely. So the idea is that when we use the DNA molecule and if we use the required amount of EcoR1, then the EcoR1 will digest the DNA completely. But on the other hand, if we use the same DNA and we add the EcoR1 restriction enzyme but in less concentration, then apart from these fragments that are generated in the complete digestion, same fragments, some other fragments will also be generated. Because the amount of EcoR1 is less, so some EcoR1 sites will not be cut from here to here. Some molecules will be generated from here to here. Some will be generated from here to here. So different sizes of fragments will be generated. So in partial digestion, that is opposite to the complete digestion, so many of the fragments will be generated. So all the possibilities of fragments will be present in this partial digestion. So scientists has the many different tools to measure the size of the fragments. In case of, so this is the technique called as gel electrophoresis, and by using this technique scientists can measure the sizes of fragments. So in case of complete digestion, we have seen in the last slide, so two fragments will be of two sizes and two fragments were of three sizes. So this is a technique in which we use some known fragments of known size, that is called as ladder. We know that this is a ladder and they have the specific sizes, for example 10 kb and 5 kb, 4 kb, 3 kb, 1 kb, 500 base pairs and so on. And in other wells, the scientists run the product of the complete digestion. So in complete digestion, they will find two bands. One will be at location 2 and we compare it here, and one will be at 3 because the two bands will overlap with each other. But in case of partial digestion, because apart from these two types of bands, there were other types of bands that have a size of 3, 4, 5, 7 and so on. So if they run in the well the product of the partial digestion, many more bands will be appear in the gel. So they can find the sizes and the number of the fragments by using this electrophoresis.